Hello, my dear students. The topic we are dealing with, that is molecular base of inheritance. Molecular base of inheritance में हम ह्यूमन जीनोमी प्रोजेक्ट पढ़ रहे हैं, ह्यूमन जीनोमी प्रोजेक्ट HGP, जिसमें हमने अभी तक पढ़ा कि इसके गोल्स क्या हैं, ये कब वजूद में आया, किस किस मुल्क ने इसका साथ लेकर इसका प्रिंट दिया, तो उसके गोल्स भी पढ़े। अब इसकी मैथडोलॉजीज़ में क्या है, देयर आर टू अप्रोच, ये आपके स्क्रीन पर नहीं आता होगा, ये मैं आपको ऐसे बताऊंगा। देयर आर टू अप्रोच फॉर सीक्वेंसिंग ह्यूमन जीनोमी: expressed sequence, tags, ESTS, identify all the genes that are expressed as RNAs and sequence of the same, and sequence of the amino. The whole genome, including both coding and non-coding regions, is first sequenced. Later functions are assigned to the different regions. In human genome project, sequence annotation has been carried out. So जो हमारा इस वक्त का टॉपिक है, आज का टॉपिक है, दैट इज कौन कौन सी मेथडोलॉजी इसमें इस्तेमाल होती है। नंबर फर्स्ट, कैसे की जाती है ह्यूमन जीनोम प्रोजेक्ट में, कैसे काम किया जाता है। द होल डीएनए ऑफ हिज सेल वाज आइसोलेटेड, यानी एक सेल का जो डीएनए होता है वो आइसोलेट किया जाता है। देन डीएनए वाज ब्रोकन इनटू रैंडम फ्रैगमेंट्स ऑफ रिलेटिवली स्मॉलर साइज। उसके बाद इसके फ्रैगमेंट्स, इसके हिस्से किए जाते हैं जो छोटे छोटे साइज के होते हैं। द फ्रैगमेंट्स वायर सेपरेटेड, द फ्रैगमेंट्स वायर अटैच्ड टू द वेक्टर्स लाइक BAC यानी बैक्टीरियल आर्टिफिशियल क्रोमोसोम्स एंड YAC yeast artificial chromosomes. They were cloned in the bacteria of the yeast cells. Alternately, the fragments could be amplified through PCR in a polymerase chain reaction. The fragments were sequenced by using automated DNA sequence developed on the principles of a method developed by the Frederick Sanger Nobel Prize for it, as well as the earlier Nobel Prize for the sequencing insulin. Then, the sequences were then arranged with the help of some overlapping fragments. The alignments of the sequence was carried out with the help of special computers-based programs. The sequences were later annotated and assigned to the individual chromosomes, 22 autosomes and Y chromosomes. The latest annotation was for chromosome first. It was completed in May 2006. Assignment of genetic and physical maps of the genome was carried out with the help of information on polymerization of restriction endonucleosides and microsatellites or repetitive DNA sequences. तो इसके बाद ये इस पर थोड़ा प्रोसेस चला। इसके बाद हम देखेंगे कि व्हाट आर द साइलेंट फीचर्स ऑफ ह्यूमन जीनोमी। Revolution of HGP. Human genome contains around about 3.1647 billion pairs of billion base pairs. Human genome project जो है इसमें कितना है बेस पेयर्स, नाइट्रोजन बेस या इसका डीएनए जो है कितना है इसमें 3.1647 बिलियन बेस पेयर्स। अगर हम फ्रूट फ्लाई ड्रोसोफिला मेलनगोस्टर हैज़ 132 million base pairs, are roughly 1/224 of total length of human genome. Number second, the total number of genes is about 21,000 protein codings, 9,000 non-coding RNA genes, and more than 21,000 pseudogenes. The number is same as present in mouth, double the number in fruit fly, and about 6 times in genes contained in Escherichia coli. Previously, it was believed that human beings carry 80,000 to 140,000 genes, 140,000 genes. It is believed that human beings carry. The pure human genomes are of no disadvantage, as most of them can code for 2 to 3 proteins by alternate splicing. Number third, the size of genome or number of genes is unconnected with the complexity of body organization. Lily has 18 times more DNA than human genome. In spite of the fact, like Lily produces fewer proteins than ह्यूमन बींग्स। इसी तरह से द फंक्शन ऑफ ओवरऑल 50% ऑफ द डिस्कवर्ड जीन्स इज अननोन। Less than 20% genome contains information for protein synthesis. By differential policing, a gene may code for 2 or more proteins. इसी तरह से part of DNA which contains repeated sequences called satellite DNA. Satellite DNA is of several types. One of them is moderately repetitive sequence 150 to 300 base pairs. They include transposes and ALU elements. There are several thousand ALU repeats in the human genome. Many satellite sequences are 11 to 60 base pairs, long hypervariable repeat sequences. They are V and TRS used in DNA fingerprinting. Over a million copies of sample sequence repeats SSR or microsatellites occurs in the genome. There are 5 to 8 pairs. The sequences are clustered around the centromere and other areas of the constitutive heterochromatin. These repeat sequences are called Jonkid DNA. Repeated sequences have no direct coding functions. They, however, provide information about chromosome structure, dynamics, and evolution. Then, point number 8, 99.9% of DNA of human population is identical, so that genomically speaking, all human beings are exactly the same. ये बड़ा अच्छा point है इसमें. This point should be noted or remembered that what is that? Silent features of human genome में हम एक point को अच्छी तरह याद रखेंगे, वो है 99.9% of the DNA of human population is identical. 99.9% of human population is identical, so that genomically speaking, all human beings are exactly same. The genetic difference is shared by various ethnic groups or races found in the various geographical regions. The genetic difference is shared by the various ethnic groups or races found in the various geographical groups, and variability that allows us to differentiate between Ram and Sham or Sita and Gita is 0.1% of the genome. Number 9, the length of the different human genes vary widely. The average size is 3000 BP. In beta globin or insulin genes are less than 10 kilobase pairs long. The longest gene of human body is that of Duchenne muscular dystrophy or X chromosome. It is 24,000 kilobase pairs long. The smallest gene is transfer RNA gene with 76 nucleotides. The halotodrenic genes SRY present on the Y chromosome is 5 kb long. It produces a 204 amino acid proteins called TDF or test determining factor. Number 10. Chromosome Y carries the minimum number of genes 231. Chromosome first has the most genes of 2968. Last point of the silent feature of human genome, what is that? About 1.4 million single base pairs DNA differences, single nucleotides, polymorphism, SNPs, pronounced as NIPS, have been identified in humans. Their number may be more than 10 million. This will be helpful in functioning chromosome location with the disease associated with the sequence of tracing human history. This was whole about the silent features of human genome project. इसके बाद इसमें हमें थोड़ा और पढ़ना है. What is that? That is DNA fingerprinting. DNA fingerprinting. वो हम कल पढ़ेंगे next lecture में. We will deal with the DNA fingerprinting. Thank you for watching.